Usually when we evaluate a medical diagnosis, there is this baseline expectation of structural precision, right? Like you look at an X-ray of a fractured radius and you see that jagged white line, and well, the structural failure is just obvious. And we have historically viewed solid tumors through a similar, I guess, somewhat binary lens.
Right, like they're just these sterile, rogue lumps of aberrant human tissue.
Exactly. But what if we completely flipped that visualization for a second? What if we viewed these tumors not as isolated masses, but as these bustling microscopic cities, complete with their own highly specialized flora and fauna?
I mean, it forces a total paradigm shift in oncology, because you are no longer dealing solely with mutated human cells.
Yeah.
You're looking at a deeply integrated interspecies ecosystem.
And for you, the learner joining us today, that is our mission for this deep dive. We're analyzing this massive 2025 review article from the journal Cell, Death, and Disease.
It's a fantastic paper.
It really is. The goal here is to understand how the intratumoral microbiota, so the bacteria, fungi, and viruses actually dwelling inside cancer cells, are acting as master architects of the tumor microenvironment.
And how they're secretly dictating whether cutting-edge immunotherapies succeed or fail.
Right. Okay, let's unpack this, because I need to establish the baseline density of these microbial cities first. Like, we know they aren't sterile, but what is the actual scale of the microbial real estate we're talking about here?
Well, the sheer volume and specificity of this microbial real estate, it depends entirely on the organ system. But the baseline numbers are just staggering.
Like, how staggering?
So recent multi-omic analyses show that up to 76% of pancreatic ductal adenocarcinoma cases, so that's PDAC, contain verifiable bacterial DNA.
76%. Wow.
Yeah. And breast and bone cancers, they consistently show bacterial signatures in over 60% of cases.
Okay, but what about other types? Is it high across the board?
No, actually. Conversely, when you sequence melanomas, that signature drops to about 14.3%.
Huh. So that tissue specificity suggests these microbes aren't just, opportunistically wandering in, right? They are being actively selected.
Precisely.
It's almost like different cancer types are these exclusive nightclubs, and they're attracting a highly specific VIP crowd based on the metabolic environment.
I love that analogy, yeah.
Because the sources highlight that lung cancer tumors are specifically enriched with microorganisms capable of metabolizing tobacco-derived compounds.
Right, so the local microbial ecology is shifting to actually consume the specific chemical byproducts of the host lifestyle.
Which is wild. So the metabolic draw is a major factor.
It is, and the exclusivity of that environment becomes even more pronounced when we look at their spatial distribution. Like, a massive portion of these populations are intracellular.
Wait, meaning they are inside the cells themselves?
Yes, exactly. They have bypassed the extracellular matrix entirely and are surviving directly inside the tumor cells.
So they are embedded directly in the structural foundation of the tumor.
Yeah. And their complexity scales with the malignancy, too.
Mm-hmm.
As the tumor progresses, you see bacteria like Thermus and Legionella becoming highly abundant in advanced and metastatic stages.
Oh, wow.
So the microbial diversity is actively scaling alongside the tumor's growth.
Okay, before we move to the next layer of this ecosystem, let's just consolidate this for you listening. We basically have 3 foundational points here.
Right, let's recap.
First, solid tumors host highly specific, often intracellular microbial ecosystems. Second, the density varies drastically by cancer type. So 76% in PDA versus just 14% in melanoma.
And third, this microscopic population scales and diversifies as the tumor advances.
Exactly. So here's a question to keep in mind. If they are embedded deep inside these cells, what is their biochemical function? Because they clearly aren't innocent bystanders, right? They must be actively modifying the cancer's survival strategy.
Oh, they are. They're actively modifying the entire local immune response. And they demonstrate this really profound dual nature.
Meaning they can help or hurt.
Basically, yeah. Within the tumor microenvironment, these microbes can function as natural immune adjuvants, meaning they actively flag the tumor for destruction.
Okay, that sounds like the good side.
Right. But alternatively, they operate as potent immunosuppressors, throwing up these biochemical shields that completely neutralize our immune cells.
Okay, if they're neutralizing the immune system, we need to break down the mechanism there. Because systemic immunotherapies are engineered to unleash these heavy cytotoxic immune responses.
Oh, absolutely.
So how can a single bacterial colony surviving inside a tumor physically halt that kind of pharmacological onslaught? I mean, without a highly targeted intervention, that shouldn't be possible.
Well, the intervention is entirely biochemical. and it operates right at the receptor level.
Give me an example.
Let's consider Fusobacterium nucleatum, or FN for short. In many contexts, FN acts as a highly effective biological shield for the tumor by secreting this specific protein called FAP2.
Okay, FAP2, and what does that do?
Well, as we know, natural killer cells, NK cells, are crucial first responders in the immune system, and they carry an inhibitory receptor on their surface called TGEAT.
So the bacteria is producing a protein that acts like a falsified biological ID badge to swipe into that receptor.
That is the exact mechanism, yeah. The FAP2 protein secreted by MT binds directly to the TGEAT receptor on the natural killer cells.
Man.
And this binding event initiates an inhibitory signaling cascade inside the NK cell. It essentially paralyzes its cytotoxic functions.
So the bacteria forces the very cells designed to destroy the tumor into a state of anergy. They just go to sleep.
Pretty much. The efficiency of it is unsettling.
It really is. But you mentioned a dual nature, which means we also have beneficial counterparts, right? Like microbes performing the opposite function.
We do. Bifidobacterium, for example, produces a metabolite called hippurate.
Hippurate, okay.
And instead of paralyzing NK cells, hippurate actually alters their metabolism to supercharge their cytotoxic capacity.
Oh, that's awesome.
Yeah, and that's been shown to drive tumor regression in melanoma models. And then we have Akkermansia mucinifila, or AKK, which produces a molecule called CDI-AMP.
Okay, if AKK is secreting CDI-AMP, it must be targeting a specific innate immune pathway, right?
Spot on. It targets the STING pathway, the stimulator of interferon genes.
Right.
When CDI-AMP binds and activates STING, it initiates this massive production of type I interferons.
Which does what exactly? Let's break that down for the listener.
Sure. So it completely reprograms the tumor microenvironment. It transforms it from an immunologically cold tumor, one that ignores therapies, into a hot tumor that's heavily infiltrated by active T cells.
Wow. Okay. So if AKK is triggering the STING pathway and calling in all these systemic immune cells, we need to look at who is actually responding to that localized environment.
Right. Particularly the tumor-associated macrophages, or TAMs.
Yes. And for you listening, let's use a visualization here to remember this macrophage polarization. Think of it like this. We have M1 macrophages, and we can call them the good cops. They are the aggressive, pro-inflammatory phenotypes that actively phagocytose, or basically eat the tumor cells and present antigens.
And then we have the M2 macrophages.
Right, the corrupt guards. They're the immunosuppressive phenotypes that promote tissue repair and angiogenesis. They effectively guard the tumor.
Exactly. And the microbes are the ones actually steering this polarization. They decide if the macrophages act like good cops or corrupt guards.
How are they doing that? What's the steering wheel?
They steer it primarily through toll-like receptors, or TLRs, right on the macrophage surface.
Okay.
Going back to our immunosuppressive example, Fusobacterium nucleatum interacts heavily with TLR4. This binding upregulates signaling pathways that force the macrophage into that protective M2 corrupt guard phenotype.
So TLR4 is the corrupting switch.
Right. Conversely, microbial D-lactic acid targets entirely different receptors, specifically TLR2 and TLR9.
And binding to TLR9 changes the macrophage's internal programming back.
Yeah, when D-lactic acid binds to TLR9, it activates the NF-kappa B signaling pathway inside the macrophage.
Okay, getting into the alphabet soup here.
I know, but it's crucial. That pathway initiates a sleeping transcriptional change. It shifts the macrophage's gene expression away from that wound-healing M2 behavior and forces it to express aggressive, pro-inflammatory M1 cytokines.
It essentially rewires the cell's operating system.
What's fascinating here is just the sheer variety of communication methods the bacteria employ to do this. They don't just rely on direct receptor binding.
Oh, what else are they doing?
They produce outer membrane vesicles, or OMVs. These act as microscopic payload delivery systems filled with bacterial DNA and proteins.
So they're shipping out packages.
Exactly. And these OMVs can travel through the tumor stroma to alter human immune cells from a distance. And on top of that, they're releasing circulating metabolites.
I saw this in the paper. Trimethylamine N oxide, or TMAO, is a prime example of that, right?
Yes. TMAO modulates inflammatory signaling cascades in nearby immune cells. We also see high production of short-chain fatty acids, or SCFAs, like butyrate.
And butyrate isn't just an energy source, right?
No, not at all. They function as histone deacetylase inhibitors, meaning they physically alter the chromatin structure inside the T cells, opening up specific genes that ramp up their cytotoxic activity.
This dense web of chemical signaling leads to a glaring clinical question, though.
Let's hear it.
If we map this out and we identify that, Fusobacterium nucleatum is using FAP2 to paralyze NK cells and TLR4 to corrupt macrophages, the immediate logical step would be to just administer broad-spectrum antibiotics, right? Clear the microbial interference and then administer the immunotherapy.
You would think so. But if we connect this to the bigger picture of tissue ecology, we hit what researchers call the context paradox.
It's context paradox.
Yeah, because biology rarely offers a universal good or bad classification. The net effect of a microbe is intrinsically tied to its spatial and genetic neighborhood.
Meaning the specific tissue microenvironment of the organ literally changes the downstream signaling of the bacteria.
Exactly. Let's track our friend Fusobacterium nucleatum across 2 different environments to see this.
Okay, let's do it.
In esophageal squamous cell carcinoma, FN drives aggressive treatment resistance. It upregulates the PD-L1 immune checkpoint and severely depletes T-cell infiltration. It's really bad news.
Right.
However, in oral squamous cell carcinoma, clinical data shows FN correlates with an improved patient prognosis.
Wait, the exact same bacteria?
The exact same one. The unique cytokine profile and immune landscape of the oral mucosa alter how the host immune system processes the FN signals. It ultimately leads to tumor suppression instead of protection.
That is just, wow. So tissue location is a massive variable, and the genetic architecture of the tumor itself adds another layer of complexity to this, right?
Oh, definitely.
The sources outline this crucial clinical algorithm based on MSI microsatellite instability. Let's explain that for the audience.
Sure. Microsatellite instability is basically a marker of a tumor's hypermutability, usually because it has a defective DNA mismatch repair system.
So it's making a ton of genetic typos.
Exactly. A tumor can be categorized as MSI high, meaning it carries a massive mutation burden and generates a ton of neoantigens. Alternatively, it can be MSS microsatellite stable, meaning it has a relatively low mutation burden.
And the microbial impact hinges entirely on this genetic baseline.
It does.
Because in colorectal cancer with an MSI high status, Fusobacterium nucleatum acts as an immunosuppressor, right? It halts immune infiltration.
Yep.
But in MSS colorectal cancer, that exact same bacteria paradoxically enhances the recruitment of T cells.
It's wild. The underlying mutational burden of the cancer cell dictates whether the microbe's biochemical signaling helps or hinders the therapy.
So the clinical algorithms of the future will literally have to sequence the host tumor genetics and the microbial genome simultaneously just to predict therapeutic efficacy.
They will have to.
Okay, let's pause and synthesize this second layer of the discussion for you listening, because that was a lot of dense immunology.
Quick 5-minute recap.
Exactly. First bullet point. Microbes talk to immune cells via complex signaling mechanisms from FAP2 proteins to OMVs and epigenetic modifiers like scFAs.
Right.
Second, they actively dictate macrophage polarization, forcing them into either those aggressive M1 good cops or immunosuppressive M2 corrupt guards via coal-like receptors.
And third, and perhaps most importantly, we have the context paradox. A microbe's impact is completely contingent on the tissue location and the tumor's mutational burden, like its MSI status.
Right. So just think about how this multivariable reality challenges the traditional generalized use of antibiotics in oncology.
It demands a level of precision we are really only just beginning to develop.
But here's where it gets really interesting. Everything we just dissected, that only covers the bacterial component.
Ah, yes.
The review article dedicates significant analysis to the microbiome and the virome, so the fungi and viruses operating within this exact same space.
The virome data is particularly revealing. When researchers run deep sequencing on the viral particles inside tumors, they find that an overwhelming majority, up to 98% in some analyses, are actually bacteriophages.
Wait, so viruses that specifically hunt and infect bacteria, not human cells?
Exactly. This reveals a hyper-complex, multi-kingdom food web operating inside the tumor microenvironment.
A microscopic food web?
Yeah. You have these bacteriophages constantly lysing or bursting open bacterial populations, and that releases bacterial DNA and proteins into the tumor stroma, which then triggers host immune responses in this continuous dynamic loop.
And the fungal populations are actively exploiting this chaotic environment too, right?
Very much so.
Because the clinical data on specific fungal culprits is pretty definitive. Like, in breast tumors, the presence of the fungus Malassezia globosa is tightly correlated with low overall survival rates.
And it's not just breast cancer. In pancreatic cancer models, Candida albicans actively colonizes the tumor and initiates this cytokine cascade.
Right. It specifically forces the cancer cells to release Interleukin-33, or IL-33.
And that IL-33 secretion acts as a targeted chemokine. It heavily recruits those immunosuppressive M2 macrophages, the corrupt guards and regulatory T cells we discussed earlier, effectively fortifying the tumor's defenses.
Which is terrifying. But you know what else stood out to me? The implications of our external lifestyle on this internal ecology are massive.
Oh, the smoking data.
Yes. The data sets in the review show that the intratumoral fungal communities are significantly richer and more diverse in the tumors of smokers compared to non-smokers.
It's a profound connection.
We aren't just causing direct DNA damage through inhaled carcinogens. We are altering the entire metabolic soil of the lung, cultivating a completely different and often more aggressive fungal ecosystem.
The macroenvironment we expose ourselves to directly selects for the microenvironment within our tissues. It's a feedback loop.
Okay, so given this incredibly dense, multi-kingdom ecosystem that governs immunotherapy response, we have to look at the clinical translation. Because, how are researchers currently exploiting these mechanisms?
Right. How do we use this?
The sources detail 3 primary clinical strategies to weaponize the microbiome. The first one is activation.
Right. Activation relies on introducing specific microbial strains to sensitize an otherwise cold tumor.
Like waking up the immune system.
Exactly. We've actually used the BCG vaccine, which is a weakened strain of Mycobacterium bovis, for decades to trigger local immune responses in bladder cancer.
Oh, interesting.
And current trials are advancing this by using oral administration of specific bifidobacterium strains to systematically enhance the efficacy of immune checkpoint inhibitors, like PD-L1 blockades.
So they're essentially priming the systemic immune system before removing the tumor's brakes.
Precisely.
Okay, so the second strategy leverages the unique physiology of solid tumors. They call it Trojan horses. And the sources highlight the intratumoral injection of engineered Clostridium novium NT spores.
This is straight out of science fiction.
It really is.
So this exploits the hypoxic or severely low oxygen cores found in rapidly growing solid tumors. Because the chaotic vascular network of a tumor just can't deliver enough oxygen to the center, it creates this necrotic core where human immune cells literally cannot survive.
Right, they suffocate.
But Clostridium, however, is an obligate anaerobe. It only survives in environments without oxygen.
It thrives precisely where our immune system fails.
Yes. So by injecting dormant spores into the tumor, they circulate until they reach that hypoxic core. There, they finally germinate, rapidly multiply, and begin lysing the tumor cells from the inside out.
Wow.
This massive cellular destruction releases a flood of tumor neoantigens, essentially ringing a massive dinner bell that draws a systemic T-cell response to a formerly inaccessible area.
It is literal biological sabotage. I love it. All right, the third strategy integrates high-tech imaging and remote control mechanisms. The review details the use of S@DIGGVs, which is a mouthful.
It is, but the tech is incredible. These are highly engineered bacteria outfitted with acoustic reporter genes, making them visible under standard ultrasound imaging.
Okay, so you can see them on a monitor.
Right. Clinicians can introduce these bacteria, visually track their accumulation within the tumor architecture, and then use focused ultrasound waves to physically trigger the bacteria.
Like a localized acoustic detonation.
Exactly. The ultrasound triggers the bacteria to release encapsulated chemotherapeutics, like doxorubicin, directly into the acidic tumor microenvironment.
Which maximizes local toxicity while minimizing those awful systemic side effects.
You hit the nail on the head.
Let's do our final synthesis here for the audience. We are no longer passive observers of the intratumoral microbiota.
Not at all.
We are weaponizing it through systemic activation, utilizing obligate anaerobes as Trojan horses in hypoxic. We have smart biological therapeutics.
We do.
Which logically leads to the reality check.
Right.
If we possess the technology to engineer ultrasound-responsive drug-delivering microbes, why is this not the standard of care in every oncology ward today?
Well, this raises an important question regarding the severe logistical and analytical limitations we currently face. The most fundamental roadblock is just the physical biomass of these microbial communities.
Because we are talking about an incredibly low biomass environment, right? Like the human gut contains trillions of microbes, but a solid tumor might only harbor a few hundred to a few thousand bacterial genomes per million human cells.
Exactly. And because that biomass is so vanishingly small, the signal-to-noise ratio in the laboratory is a massive hurdle. Contamination is a persistent daily threat.
Like how sensitive is it?
A single degraded bacterial fragment in a sterile chemical reagent or microscopic environmental dust just floating into the sequencing machine can completely drown out the authentic microbial signature of the tumor biopsy.
That's incredibly frustrating for researchers, I imagine.
Oh, it is. And the sequencing technologies themselves present a trade-off. We have 16S RNA sequencing, which is cost-effective and widely used, but it lacks resolution. It can identify the broad family or genus of the bacteria, but it cannot differentiate between specific strains. And more importantly, it cannot tell us what genes those bacteria are actively expressing.
So you don't know what they're actually doing.
Right. To get that functional data, we need whole genome sequencing, or WGS. WGS provides strain-level resolution and maps the entire metabolic potential of the microbe.
So why not just use WGS for everything?
The high costs and the extreme sensitivity to the human DNA background make it incredibly difficult to scale across large patient cohorts.
Got it. Furthermore, we are constantly battling the correlation versus causation loop, right? We observe an abundance of Malassezia globosa in a highly aggressive breast tumor, but we have to prove causality. Did the fungus actively rewire the microenvironment to make the tumor aggressive? Or did the aggressive mutation profile of the tumor simply create a highly favorable metabolic niche for that specific fungus to opportunistically colonize?
That's the million-dollar question. And snapshots in time cannot answer that. The field requires complex spatial temporal tracking.
Meaning tracking them through space and time.
Right. We need to monitor human patients over time to map exactly where the microbes are localized within the tumor architecture and how their gene expression actively shifts the literal moment a pharmacological intervention is introduced.
It's a huge undertaking. So, what does this all mean? Let's bring the core insights of this deep dive together for you listening. We started by dismantling the outdated view of tumors as just these sterile masses. We've explored them as deeply complex, multi-kingdom ecosystems. The microscopic life inside them, the bacteria secreting FAP2, the fungi initiating IL-33 cascades, the bacteriophages modulating the entire food web, they act as the ultimate biological middlemen.
They really do.
They actively negotiate with our immune system, polarizing macrophages and suppressing NK cells, and ultimately dictating whether a patient's immunotherapy can breach the tumor's defenses.
Understanding and manipulating those complex signaling networks represents the absolute next great frontier in targeted oncology.
I agree completely. And I want to leave you, our listener, with a final provocative paradigm shift to consider. If a malignant tumor is not merely a mass of rogue human cells, but a vibrant, highly interactive interspecies ecosystem, how does that redefine the role of an oncologist?
That's a great question.
Does the future of medicine require us to move away from simply being cancer killers, deploying cytotoxic drugs, and instead become cellular ecologists? Will the protocols of tomorrow focus less on eradicating human tissue and more on precisely managing the microscopic wildlife within it to just let our own immune system do the work?
It's entirely possible.
Review the mechanistic data on macrophage polarization, consider the impact of mutational burden algorithms, and just think about how this changes your fundamental understanding of human pathology. Until next time, keep analyzing the data and keep diving deep.